Thanks for joining us in another fascinating journey through the world of science here on the Science Channel podcast.
It's great to be diving into another topic.
If you're finding these explorations intriguing, please do give us a like and subscribe. It really helps support what we do.
Absolutely. Every bit helps.
Okay, so today we're tackling a real biological riddle. It involves, bats.
Ah, yes, the masters of the night sky.
Exactly. And the question is, how do these remarkably long-lived little mammals seem to sidestep cancer so effectively?
It's a genuine puzzle, isn't it? Especially when you think about things like, well, their high metabolism during flight.
Right, and all the viruses they seem to carry without getting sick, factors that you'd normally associate with higher cancer risk.
Precisely. We're talking about creatures where, many species live for decades, 20, 30, even over 40 years for some.
Which is just incredible for their size.
It really is. There's this metric called the longevity quotient, or LQ. Basically, it compares actual lifespan to what you'd predict based on body mass.
And for bats?
Well, for about 65 species studied, the average LQ is around 3.5, meaning they live, on average, 3.5 times longer than expected.
Wow. And normally in biology, longer life means more cell divisions, more chances for
mutations, for DNA errors to build up, more exposure to stressors. So yeah, usually longer life equals higher cancer risk.
But bats seem to break that rule.
They really do. Reports of tumors, whether in wild bats or those in captivity, are surprisingly uncommon. It's a fascinating paradox.
Okay, so let's try and unpack this. What's going on inside their cells? We're going to dig into some really cool new research today.
Yeah, this research looked closely at the cells themselves, specifically fibroblasts.
Those are the connective tissue cells, right?
That's them. They studied these cells from 4 different bat species, chosen to kind of span the bat evolutionary tree.
And the mission here is to understand what this research reveals about their unique defenses against cancer based on a key scientific article.
Right. Researchers generally look at cancer resistance from 2 angles. There's the cell autonomous stuff.
Meaning things inherent to the cell itself.
Exactly.
Yeah.
Intrinsic properties. And then there's non-cell autonomous. How cells interact with their environment, the immune system, things like that.
We've seen examples in other long-lived animals, haven't we?
Oh, definitely. Like elephants have extra copies of tumor suppressor genes like TT53. Some rodents have super efficient DNA repair. Large animals often regulate telomerase differently.
But studying this kind of thing directly in bat cells, trying to actually make them cancerous in the lab, that's been more limited.
Until fairly recently, yes. There wasn't a lot of experimental work on transforming bat primary cells.
Okay, so this new research dives right into that, and they looked at something called oncogenic hits.
Yes, basically, how many specific genetic changes or hits to key pathways you need to force a normal cell to become cancerous in a lab dish.
And how does that usually compare?
Well, for human cells, it generally takes about 5 or 6 hits. For mouse cells, interestingly, it's only 2.
Only 2 for mice. Okay. And the bats.
This is where it gets really surprising. For 3 of the bat species, they studied the little brown bat, the big brown bat, and the cave nectar bat.
Yeah.
It only took 2 2-0 hits, just like the mice.
Wow. So activating one gene, HRAS, and knocking out either P53 or PRB,
Right.
That was enough.
That was enough to transform their fibroblasts in the lab. They even saw the same thing in kidney cells from the big brown bat.
And this isn't just a one-off finding.
Doesn't seem like it. There was another recent study looking at 7 different Asian bat species, and they reported something very similar. It suggests this ease of transformation might be, well, pretty widespread among bats.
Okay, hold on. If their cells are so easy to transform in a dish, why aren't they riddled with cancer in the wild?
That's the million-dollar question, isn't it?
Yeah.
It strongly implies they must have other very potent mechanisms protecting them in vivo in the actual animal.
So maybe it's not about stopping the initial transformation, but something else.
Potentially. Or maybe there are multiple layers of defense. One area they looked at was telomerase.
Right, the enzyme that maintains the ends of chromosomes. In big mammals like us, it's usually shut down in most body cells.
It's considered a tumor suppressor mechanism, limiting the number of times a cell can divide.
But in these bats,
They found that all 4 species they studied had active telomerase in their wing fibroblasts, and in other tissues too.
active telomerase, so their cells can potentially keep dividing for longer.
It seems so. Consistent with that, the bat fibroblasts didn't show replicative senescence in culture. They just kept proliferating.
Which again sounds kind of pro-cancer, doesn't it?
On the surface, yes. It's another trait that in many other species would be seen as increasing risk. They also noticed smaller bats tended to have higher telomerase activity, which fits a known pattern.
So, easy transformation, active telomerase. It sounds like bats are playing with fire. What else did the researchers investigate?
They looked at senescence again, but this time, stress-induced premature senescence, or SIPS.
That's when cells shut down early due to stress, like radiation, acts as a barrier to cancer.
Exactly. It stops damaged cells from multiplying. But it also contributes to aging over time.
And what did they find in the bat cells?
Well, compared to human and mouse cells, fibroblasts from the little brown bat and the Jamaican fruit bat showed significantly less SIPS after radiation exposure.
Less shutdown.
Less shutdown. And when the bat cells did become senescent, they behaved differently too.
How so?
They produced lower levels of many of those inflammatory molecules that senescent cells usually secrete the SASP factors.
SASP, Senescence Associated Secretory Proteins. Those can cause problems for neighboring tissues, right? Promote inflammation.
That's right. The bat cells, especially from the little brown bat, had a much more muted inflammatory response. Lower interferon signaling, for example.
Okay, so less likely to shut down from stress and less inflammatory if they do. But there's a flip side.
A very important flip side. Those same bat cells showed a significant increase in apoptosis programmed cell death when hit with radiation. Much more than mouse or human cells.
Ah, so instead of just becoming senescent and maybe causing inflammation, they're more likely to just self-destruct if damaged.
Precisely. And this seems to be driven by the p53 pathway. It looks like enhanced apoptosis could be a really key anti-cancer strategy for them.
p53, the guardian of the genome. Makes sense. So did they find anything special about p53 activity in bats?
They did. Bat fibroblasts generally showed higher baseline p53 activity compared to human and mouse cells.
Just ticking over at a higher level normally?
Seems like it, especially in the little brown bat and big brown bat.
Yeah.
And when they used a drug, Nutlin-3, which stabilizes p53.
Let me guess, more apoptosis.
Significantly more apoptosis in the bat cells compared to mouse and human cells.
And at the gene level.
RNA sequencing showed higher levels of the TP53 gene transcript itself in untreated bat cells. And after radiation, you saw upregulation of p53 target genes involved in apoptosis, like BAX, and also genes that halt the cell cycle.
So the whole system seems dialed up.
Dialed up and maybe finely tuned. They also saw upregulation of genes that regulate p53, like MDM2, which inhibits it, and WRIAP53, which stabilizes it. It's just, complex control system.
And didn't they also find something about DNA repair genes?
Yes. Briefly, they mentioned evidence for positive selection on DNA repair genes in bats generally, suggesting evolution has favored keeping their DNA in good shape.
Okay, so enhanced p53 activity seems crucial. And this links back to those elephants you mentioned earlier, right, with the extra gene copies.
Exactly. Elephants have multiple copies of TP53, which is thought to contribute to their cancer resistance. So the researchers looked at TP53 copy number in the bats.
Yeah.
Several bat species are known to have 2 to 4 copies. But get this. The little brown bat, Myotis lucifugus, appeared to have 7 copies in its genome. 7. And the big brown bat, Eptesicus fuscus, had 3. Further analysis suggested at least 2 full-length functional copies in the little brown bat.
So that extra gene dosage could really ramp up the p53 response.
It certainly could contribute significantly to that enhanced apoptosis we were talking about. And even in bats that don't have extra copies of the gene itself.
Yes.
They found evidence for a higher number of promoters for the TP53 gene. Those are the regions that control how much the gene is turned on.
Ah, so even with 1 gene copy, they might just make more p53 protein.
Potentially, yes. It looks like bats might have evolved multiple ways to boost their p53 defenses.
Okay, so let's try and tie this together. We've got cells that seem quite easy to transform in the lab. They have active telomerase, which usually allows more cell division.
Right.
They have a dampened stress-induced senescence and inflammatory response. But they have this incredibly souped-up p53 system ready to trigger cell death at the first sign of serious trouble, possibly boosted by extra gene copies.
That seems to be the picture emerging from the cellular level, yes.
So, given the easy transformation in vitro, does this suggest that maybe the in vivo protection relies on more than just these cell-intrinsic mechanisms?
That's the leading hypothesis now. Considering everything the easy transformation, the telomerase, it seems likely that bats rely heavily on non-cell autonomous mechanisms too, particularly a highly efficient immune system.
Immunosurveillance, the immune system finding and killing precancerous cells.
Exactly. We already know bats have some pretty unique immune system adaptations, like how they tolerate viruses often involves dampening inflammation.
Could that dampened inflammation also help them deal with those SASP signals from any senescent cells that do pop up, preventing chronic inflammation?
That's a very plausible idea. And there's some preliminary evidence pointing towards stronger immune surveillance, too. One study suggested higher expression related to natural killer cells, or NK cells, in bat skin compared to other species.
NK cells are key players in hunting down tumor cells, aren't they?
They absolutely are.
Yeah.
So a more active NK cell population could be constantly patrolling, eliminating abnormal cells before they get a chance to form a tumor.
So it's this fascinating combination then. On one hand, cells with traits that look risky, easy transformation, telomerase activity.
But counterbalanced by these potent internal defenses like supercharged apoptosis driven by p53, and maybe less damaging senescence.
And then likely layered on top of that is a really vigilant immune system cleaning up any escapees.
That seems to be the most likely explanation for their remarkable cancer resistance. They're playing by a different rule book, it seems.
It really makes you think, doesn't it? Could understanding this unique interplay in bats, this balancing act between cellular traits and immune defenses, could that give us new clues for fighting cancer in humans?
It's a very compelling possibility. What can we learn from an animal that seems to defy some of our basic assumptions about cancer development?
Lots to ponder there. A truly fascinating area of research.
Indeed it is.
As always, don't forget to like the video and subscribe if you haven't joined us yet.